about me and you. And I think to myself, what a wonderful world.
So welcome to this another Coco channel video, and now we are going to talk about the microbial genetics, which include the genetics of bacteria and the viruses. So as you can see here, I have a picture on the screen. You can see that the animal cell has a nucleus and a membrane-bound organelles, because the animal cells are eukaryotic organisms. In case of bacteria, they are very small and they don't have nucleus and membrane-bound organelles. Instead, they have the nucleoid region, which inside of this are the single cellular chromosome. So if you look at the protein synthesis between prokaryotes and eukaryotes, the prokaryotes has a much simpler, because it has the absence of nucleus and there is no transcription take place in. When the DNA is transcribed to messenger RNA, it doesn't receive any processing, so instead it goes straight to the ribosome, where the messenger RNA will be translated into a protein. So prokaryotic and eukaryotic cells are considered living because they are made up of cells, while in the case of a virus, it is not considered alive because it is not made up of cells. Instead, viruses are made up of a genetic material surrounded by a protein coat. So now we will be looking at the genetic of viruses. Here we have 4 types of viruses, which are the tobacco music virus, which are cylindrical virus and its genetic information or the RNA. And the second type of virus are the adenovirus, which genetic material are the DNA, and it causes common colds to us. And the other one is influenza virus, which cause a flu, and its genetic material are the RNA. And the last one is the bacteriophage, with genetic information are the DNA, and it only infects bacteria. So all of those 4 types of viruses, it has its own distinct shapes. So the viruses are considered a parasite. It doesn't have ribosomes and enzymes. They have a specific host range. It is literally used that cells against itself in order for the virus to reproduce. Host range means they have a particular type of cell. For example, is the hyper T cells in the HIV virus, which targets the hyper T cells, which are the white blood cells are found in our immune system. Made a drawing how the virus enter the host cell. So the virus will attach the host cell and the viral DNA enters the cell. Then this viral DNA will use the enzymes and nucleotides of this host cell to replicate itself. Then the viral DNA will come other host cell material to transcript its genes to the messenger RNA and translate the mRNA into capsid proteins. Then viral DNA and capsid proteins will assemble itself to a new virus and will leave or exit the host cell and leave the cell destroyed on the process, and those new form of viruses will continue to infect other cells. So there are 2 types of reproductive cycles that a bacteriophage can enter. One is the lytic cycle and the other one is the lysogenic cycle. The goal of the lytic cycle is to kill the host cell. The virus that undergo this lytic cycle are called the virulent virus. So I have here a picture in the screen. So when the bacteriophage lands on the bacteria, example is the E. coli, this bacteriophage will stick its fiber to the bacteria and insert its own DNA, and this will cause the DNA of the host cell to be cut off in the process of hydrolysis. Then bacteriophage uses the host cell enzymes and its ribosomes to manufacture new bacteriophages, and those bacteriophages assemble itself, and then they are going to go out and leave the host cell, burst, and eventually die. The other reproductive cycle is called the lysogenic cycle. This one, it does not destroy the host cell, and the viruses undergo the lysogenic cycle are called the temperate viruses. Once the bacteriophage attaches the host cell and inject its DNA, and the DNA is going to incorporate itself into the host cell DNA. So once the virus's DNA is incorporated to the host cell's DNA, it is now called a prophage, and this prophage is formed every time the bacteria reproduces, and it will also reproduce viral DNA every time the bacteria produce proteins, and it will also produce viral proteins. Then eventually the prophage will leave the host cell genome. Then the virus will then convert from lysogenic cycle into lytic cycle, which again going to kill the host cell. So we have here the comparison between the lysogenic and lytic cycle. So the goal of the lytic cycle is to kill the host cell, and the virus which undergo this lytic cycle is called the virulent virus, and the other one is lysogenic cycle. It doesn't kill the host cell, and the virus that undergo lysogenic cycle are called the temperate viruses. Now let's take a look at the HIV, or the human immunodeficiency virus. This is the virus which called the retrovirus because it has RNA as its genetic information. So you see here, I have a picture on the screen. It has a capsid of protein coat. Inside of this protein coat is RNA. Inside the capsid along with the RNA is called the reverse transcriptase. So now I will explain how the HIV virus will enter the cell, the host cell. So the HIV virus enters the host cell through endocytosis, and once the viral RNA has entered the host cell, reverse transcriptase is gonna reverse the translated viral RNA to the DNA. So once the DNA is inserted, it's going to enter the host cell genome and become a provirus. And this provirus is the same with the prophage, but the difference is this prophage is only found in bacteria, but the provirus is found in the eukaryotic cells. Now every time the white blood cell goes through a protein synthesis, it will manufacture a new RNA as well, which is the genetic material of the HIV virus. And it will also manufacture a viral proteins. So the newly assembled HIV virus will going to accept the white blood cell and then go through a exocytosis, and they will leave the host cell destroyed, and nuclear envelope will be formed outside. Besides the viruses, there are also simple infectional agents like a viroids, which is the circular RNA molecules that infect the plants, and the other one is prions, which are the infectious diseases. For example, is the mad cow disease. So now let's proceed to the bacteria genetics. So the important genetic structure in bacteria is the plasmid, which are the circular chromosomes. This is important because this is where the genetic recombination takes place. And also the bacteria has a high mutation forms. It has 3 forms of recombination, and they have a variation. And this genetic variation is crucial for the process of natural selection. And bacteria has also the ability to transfer this plasmid to one bacteria to another bacteria. So genetic recombination. So what is genetic recombination? It is the exchange of genetic material between one organism, which leads to a production of new copy of that bacteria or organism. Here we have the picture of a genetic combination. So when the bacteria grows in a petri dish, they are going to reproduce called the colonies. These are made up of thousands of bacteria. So there are three different methods of genetic information. One is through the process of transformation. This is where the bacteria takes in plasmids from the environment. And to do that, they must induce competency in the lab. Through the process of heat shocking, exposing the bacteria to high temperature, to low temperature, so that it will open its specialized proteins in the cell membrane to allow the bacteria take in its plasmids. So once the bacteria take in its plasmids, they can express the genes on the plasmids and they have a new trait. It can be either resistant to antibiotics. And the other one is the transduction. This is another when phages are used to transfer DNA from one bacteria to another bacteria. There are two forms of transduction. One is the generalized transduction. They are random pieces of host DNA in generalized transduction, and the other one is a specialized transduction. The prophage carries the genes. These two forms are just the same. The only difference is in generalized transduction, the phage is going to utilize the lytic cycle, and while the specialized transduction, it utilizes a lysogenic cycle. So generalized transduction, the phage will land to the bacteria and it will insert its genetic material to the host cell. Then the bacterial DNA will be cut off and new phages are created leaving the host cell. But as you can see in the picture right here, one of these phages, instead of viral DNA, it has a bacterial DNA instead. So when this phage exits the host bacteria and infects new bacteria, instead of injecting its own viral genetic information, it's injecting the DNA that come from the prion bacteria. So specialized transduction, this one utilizes the lysogenic cycle, and prophage is going to leave the bacteria DNA, except inside of the prophage there is a small piece of bacteria DNA. So when new viruses assemble itself inside one of those viruses, it's gonna be a bacterial DNA. And when that virus infects new bacteria, it is going to insert the DNA from the prion bacteria. So here's another method by which the DNA can be transferred from one bacteria to another bacteria. This is the conjugation. And some bacteria has F plasmid, and F plasmid contains genes that allow the bacteria to form what we call the sex pili. So the sex pili here is the projection allows bacteria to form a cytoplasmic bridge with another bacteria, and once the bridge is formed, the plasmid replicate and move to the second bacteria. And once this bacteria receives the F plasmids, it will allow its ability to create a new sex pili for itself. So when bacteria receive the F plasmid will then insert itself into the bacteria DNA. So when the F plasmid is inserted in the HFR cell, now can form cytoplasmic bridge with other bacteria, actively change the genetic information. Bacteria that have a plasmid and can form sex pili are called the male bacteria. And bacteria that does not have the F plasmid that cannot form a sex pili are called the female bacteria. And other plasmid is the R plasmid, which allow the bacteria to be resistant to the antibiotics.